Hey everyone, and welcome to RUBIN'S Mind Lab, where we break down the latest research into what it really means. Ever wonder how your brain goes from a tiny simple thing to the complex powerhouse it is, especially when you are a baby or a young child? Well, a fascinating new review looks at just that, exploring how our experiences shape our brains during those super important early years. Today we're diving into the world of experience-dependent synaptic plasticity in early development. That is a mouthful, I know. Let's break it down. Basically, it is all about how the connections in our brain, called synapses, change and grow based on what we experience from birth to about 6 years old. Think of it like your brain building its wiring system, and the more you use different parts, the stronger those connections become. The scientists behind this research noticed that a lot of cool studies about this topic are spread out. Some are done in labs with animals, looking at the tiny details of how brain cells work. Others are trying to connect those animal findings to what might happen in humans. And then there are studies directly observing kids. This makes it tricky to get a clear picture of the whole story. So, their goal was to bring all this information together and see what we can really learn. And importantly, what we can actually claim to know based on the type of evidence available. They wanted to figure out which brain mechanisms are involved, what things influence these changes, and how far we can confidently extend what we learn from animal studies to human babies and toddlers. So how did they do it? They performed what is called an integrative review. Imagine it like a superpowered literature search and organization project. They dug through tons of research papers published between 2012 and 2026 in two big academic databases. They were looking for studies on how experience affects brain connections, especially during critical or sensitive periods in early childhood. They ended up with a lot of research to sort through. After carefully screening out the ones that didn't quite fit, they narrowed it down to 49 articles that they considered the most relevant and high quality for their review. They then classified these studies into 3 levels based on how directly they apply to humans. Level 1 studies were those done in animals, giving us the nitty-gritty on the cellular and molecular mechanisms. Think of these as the blueprints. Level 2 studies were translational, trying to bridge the gap between animal findings and human possibilities. These are like the engineer's initial design sketches based on the blueprints. Level 3 studies were direct human evidence, mostly involving babies and young children, giving us direct observations of what is happening. These are our real-world examples and user feedback. What did they find? They identified 4 main ways our brains change based on experience during these early years. First, there is activity-dependent synaptogenesis and pruning. This means your brain actively creates new connections and gets rid of old, unused ones based on how you use it, building and then sculpting its own wiring. Second, glial regulation. Glia are like the support crew for your brain cells, and they play a surprisingly active role in shaping those connections. They're not just there to clean up, they help build and refine things too. Third, myelination. This is like adding insulation to your brain's electrical wires, making signals travel faster and more efficiently. And fourth, perineuronal. Net formation. These are like special coverings that can stabilize connections, helping to make certain brain circuits more permanent. The bulk of the research, 30 out of the 49 studies, came from animal models. These studies really helped them understand the nitty-gritty details. For example, they found that early brain activity, even spontaneous activity in a newborn's brain, helps organize neural circuits, especially in areas related to senses like vision and hearing. In the hippocampus, a key area for learning and memory, they saw that the timing and type of brain activity are crucial for synapses to mature properly. Some studies looked at specific molecules and genes. For instance, one study found that messing with a particular protein called VGD impacted how brain cells communicated, how they grew their branches, how many tiny bumps called spines they had on their surfaces, and even how well the animals could learn to navigate. Another study showed that a problem with a gene called SYNGAP1 sped up the maturation of connections, but also limited the brain's ability to change during an early critical period. These findings really highlight how complex the molecular dance is when the brain is developing. Interestingly, even things like an enriched environment in these animal studies made a difference. When young animals had more stimulating experiences, they grew more connections, had more complex brain structures, and their vision improved. This was linked to changes in genes that are involved in brain activity and learning. It really shows that use it or lose it can apply to brain wiring even at the molecular level. The role of glia, that support crew I mentioned, really stood out. Studies showed that different types of glia, like astrocytes and microglia, are actively involved in helping synapses mature, fine-tuning brain plasticity, and refining circuits in sensory and decision-making areas. They're not just passive helpers, but active sculptors of our developing brains. However, the researchers pointed out that direct human evidence on glia in babies and toddlers is still pretty limited, so we should be careful about jumping to conclusions about humans based on this. And then there's myelination. This process where nerve fibers get wrapped in a fatty sheath is like upgrading your brain's internet speed. It makes brain signals zip around much faster and helps different parts of the brain work together more smoothly, like a well-coordinated orchestra. Translational studies showed that this myelinated axon plasticity is important for how circuits form and function, and in humans, we see this progressive network integration as an infant's brain develops. They also looked at specific senses. In the auditory system, for instance, they found that not hearing properly and then getting a cochlear implant showed how important it is to intervene during sensitive periods. This means that early sensory experiences can shape brain function, but it doesn't necessarily mean the developmental process is set in stone forever. There is still room for change. Remember those perineuronal nets I mentioned? They seem to play a role in stabilizing brain circuits and contributing to the closing of those windows of heightened plasticity. In animals, these nets can limit how much connections can change, helping to lock in certain brain patterns. This suggests that when a critical period closes, it is not a complete shutdown of change, but rather a shift to more specialized and stable forms of learning. The researchers also found that adversity during pregnancy or around birth can significantly disrupt these critical windows, especially in the hippocampus. For example, studies on animals born with restricted growth or experiencing stress during development showed altered timing in the opening and closing of important brain periods related to memory. These disruptions were linked to changes in myelination and other key developmental processes. Beyond specific biological mechanisms, the review emphasizes that early experience is a broad context. Sensory input, yes, but also the quality of caregiving, like synchrony between a parent and child, and the simple act of touch are super important. These relational aspects are linked to how babies regulate their bodies and develop their social and emotional selves. On the flip side, early adversity and poverty can negatively impact development through a range of pathways, affecting stress hormones, the immune system, gene expression, and how brain regions connect. While this human evidence is mostly observational, meaning it shows associations rather than direct cause and effect, it strongly suggests these factors are playing a significant role. One notable finding from a human-relevant study was about early life stress. It was found to mess with the development of brain cells in the auditory cortex during a critical period. This made them see more of certain types of cells and increase the expression of a gene linked to important brain receptors. This indicates that adversity can impact not just emotional or cognitive areas, but even sensory processing centers during these sensitive times. The researchers also pointed out that these sensitive periods aren't the same for every skill. The timing for developing motor skills, for example, seems different from visual or language development. They found that early motor skills depend on sensing and moving, while language development is heavily influenced by early sensory input and interventions. Social-emotional growth, on the other hand, is deeply tied to caregiver interactions and a sense of safety. Crucially, direct human evidence also helps us understand that human development isn't just a copy-paste from animal models. Studies on the human visual cortex show that while there are rapid changes early on, some key proteins involved in brain connections continue to mature well into childhood. This means human-sensitive windows might be a bit more flexible and prolonged than what we see in some animal studies, not following a super rigid timetable. So to tie it all together, this review really shows that early brain development is a dynamic process shaped by our experiences. It is not just a one-time event, but an ongoing calibration. The brain builds connections, fine-tunes them, and uses experience to organize itself. And this process is influenced by everything from the sounds we hear to the way we're cared for. The key takeaway is that these critical or sensitive periods are really windows of opportunity, not closed doors. There are times when the brain is particularly efficient at learning and organizing. But this doesn't mean later development is impossible. It just means that changes might happen differently or require different kinds of experiences. The review also emphasizes that we need to be careful not to oversimplify things. Findings from animal research, while incredibly valuable for understanding mechanisms, shouldn't be directly translated into specific advice for parents or educators without further human studies. What is really great about this review is how it helps us understand the weight of different types of evidence. Animal studies give us precision about how things work at the tiniest level. Translational studies help us think about how that could apply to humans, and direct human studies show us what is actually happening in babies and kids. By sorting it this way, They help us avoid making overly strong claims that aren't fully supported by the evidence. A really important point they make is that we cannot jump from what we see in a lab rat to telling parents exactly what to do. The science is still evolving, and we need more research specifically looking at how these mechanisms play out in human children. But that doesn't mean there is no practical value. The evidence does suggest that providing good sensory experiences, stable relationships, and reducing stress for young children is really important for healthy brain development. Now, like any scientific endeavor, this review has its limitations. For one, as they mentioned, the bulk of the research was in animals, which creates a bit of a distance from human realities. Also, getting detailed cellular information directly from human babies is incredibly difficult, so definitive causal links are harder to establish in humans. Many of the human studies they reviewed were observational, meaning they can show that two things happen together but not necessarily that one causes the other. And finally, the sheer variety of mechanisms, developmental areas, and study types makes it hard to draw one single simple storyline for early brain development. In conclusion, this review paints a picture of early brain development as a flexible and experience-driven process. It is constantly being shaped by our interactions with the world. While critical periods are times of heightened opportunity, they're not rigid destiny makers. The research strongly supports the idea that early experiences are fundamental for setting up our brain circuitry, but they don't seal our fate. From my perspective as a science communicator, this is exactly the kind of nuanced understanding we need. It's exciting to know how much potential there is for positive development in early childhood, and how much nurture matters. But it is also crucial to be responsible with this knowledge, to advocate for supportive environments for children, and to remember that every child is an individual with their own unique developmental journey. Future research will hopefully continue to fill in the gaps, especially by bridging that gap between animal mechanisms and human outcomes with more direct human studies and ethical interventions. What do you think about how our experiences shape our brains from such a young age? Let me know in the comments below. If you enjoyed this deep dive into developmental neuroscience, please give this video a thumbs up. Subscribe to RUBIN'S Mind Lab for more science breakdowns and hit that notification bell so you don't miss our next episode. Thanks for watching, and I'll see you next time.